All right, class, so here we are Wednesday, about the middle of October. We're going to go over chapter 11, which is blood today. And this is usually the chapter that we go over before we start your cardiovascular system, where we'll go over your heart and all the blood vessels. And before we go over your cardiovascular system, we talk about blood and the components of blood. We'll talk a little bit about blood tests. This will be the lab kit you're picking up, will be a synthetic blood typing kit. So when we do that lab activity next week or the week after, you guys will be testing that synthetic blood for the blood type. So that'll be fun. So here we have the functions of blood. Basically, this is your body's highway system. So it transports everything: gases, nutrients, waste products, oxygen, carbon dioxide, processed molecules, regulatory molecules. It helps to regulate your pH levels and osmosis. So that's the diffusion of water. It helps to maintain your body temperature because blood carries heat. George, good question in the chat, asking if it has to be refrigerated. If there aren't any instructions when you got it, it should be okay. Otherwise, there should be specific instructions. I think this kit can just stay out. It's the next kit, the urinalysis kit, that might need some special attention, but your blood kits should be okay, I think. Great question. Room temp was okay. Thanks, Cameron. Yeah, so room temperature should be fine. It's just synthetic blood. It's not real. So yeah, it should be able to withstand. Room temperature is good. Maintenance of body temperature. It helps to protect against foreign substances because your blood includes all your white blood cells, which are all of the cells that fight off infection. And it helps to form clots because your blood has platelets in it that help to form a clot if there's ever damage to a blood vessel or another body part and it needs to stop bleeding. So here we have the components of blood. Plasma makes up about 55% of your total blood. And plasma is pale yellow in color. It's a yellow liquid that surrounds the cell. So your plasma is not any cells to it. It's just mainly water, proteins, and about 2% of other solutes, but it doesn't contain cells. Your plasma just surrounds the cells in the blood. And then the formed elements are all of the cells and the cell fragments. So the formed elements make up the other 45% of total blood. Your formed elements include all of your cells and fragments. So we have erythrocytes, our red blood cells. The leukocytes are your white blood cells, the ones that fight off infection. And then the thrombocytes will kind of separate out to create platelets, which are involved in the blood clots. Some of the proteins within your plasma are shown here. And I seem kind of choppy. I just keep letting some people in, which is no problem. So we have albumin, which is the majority of your plasma proteins. It helps to maintain your water balance. You have globulins and fibrinogen. Globulins help the immune system, and fibrinogen creates little tiny fibers that help to aid in clot formation of those platelets. So if we were to take a blood sample, and many of us have probably had a blood sample taken before, so they take blood, usually this vein in the cubital part of the elbow is the best place to take a blood sample. And a lot of you guys probably know this because you're EMTs or have medical experience. So when we take that blood sample, in order to separate out the components, we put it in a vial and then centrifuge it, which means it spins it down extremely fast. And when we do that, all the formed elements float to the bottom. So like the red blood cells, the white blood cells, the platelets are not the platelets, but the red and white blood cells are the formed elements that float to the bottom. And because they're more dense, the buffy coat makes up a little teeny tiny kind of separation between the formed elements, and then the plasma kind of floats to the top because it's less dense because it's mainly water. So here is what makes up most of the plasma: proteins, water, other solutes. Here we have the formed elements then are the platelets, white blood cells, and red blood cells. So that's the composition of blood and kind of how we can separate it out. And the plasma makes up 55% with the formed elements on the bottom making up 45%. And then the buffy coat is a little separation between the two. Hematopoiesis is the process that produces formed elements. So hemato has to do with blood and poiesis means forming it. In the fetus in the womb, hematopoiesis occurs in several tissues: the liver, the thymus, the spleen, the lymph nodes, and the red bone marrow. And after birth, hematopoiesis specifically occurs in just your red bone marrow, which occurs in your bones, but some white blood cells are produced in lymphatic tissues. All the formed elements of blood are derived from a single population of cells called a stem cell, or hematocytoblast. And this is kind of interesting. All of your white blood cells, red blood cells, and platelets all come from one cell that will differentiate to give rise to the various cell lines, each of which will end with the formation of a particular type of formed element. So here we kind of have this stem cell. This is the hematocytoblast, and it either will differentiate into the myeloid stem line or the lymphoid stem line. The lymphoid stem line just goes down and forms a lymphocyte, which is one of your 5 types of white blood cells. And then the myeloid stem line eventually goes on to differentiate into a red blood cell, which carries oxygen, the platelet, which helps in blood clot formation, or then one of these 4 leukocytes, white blood cells: the basophil, eosinophil, neutrophil, and monocyte. These 5 at the bottom make up your white blood cells, so a part of the immune response. The red blood cell carries oxygen, and then the platelets are involved in clot formation. So again, it's kind of interesting that all of these 5 types of blood cells are kind of start off as one stem cell, the hematocytoblast, which will then differentiate into those 1 of 5 formed elements. So we'll start with your erythrocyte. It looks kind of like a donut, a biconcave disc shape. The disc shaped has thick edges. The nucleus is lost during development. So mature erythrocytes do not have a nucleus. And that's because it's just full of the protein hemoglobin to transport oxygen. So it doesn't have a nucleus. Its only function is to transport oxygen. So we pack hemoglobin, which is the protein, inside the red blood cell to help transport the oxygen to our tissues. And erythrocytes live for about 120 days in the bloodstream until they go to the spleen, where they're recycled and broken down. This is a look, a scanning electron micrograph, about 2,600 times magnified. You can see the nice, beautiful red blood cells. You can see how the white blood cells look different. And then the smaller kind of cell fragments are called platelets. Hemoglobin then is the main component of erythrocytes. It's a type of protein that transports oxygen. Each globin protein is attached to a heme molecule. Each heme contains 1 iron atom, and the oxygen is what will bind to that iron in this hemoglobin molecule. If we have oxyhemoglobin, that means that hemoglobin has the oxygen attached to it. So this is the hemoglobin molecule. It's made up of kind of 4 globin chains. And you can see these chains here. They all have different names. So here's kind of 1 chain, 2 chain, 3 chain, and then 4 chain. And in the middle, the little blue circle, I'm not very good at circling these. You can kind of see the 4 heme groups. And these 4 heme groups are what will be where the iron is attached, and that's where the oxygen attaches. So 1 hemoglobin molecule can actually transport 4 oxygen molecules because of those 4 heme groups, and each heme group with each has an iron can transport 1 oxygen. So again, 1 hemoglobin molecule can transport 4 oxygens because they'll all bind to one of these 4 blue heme groups. So that's hemoglobin. Production of erythrocytes. So if we have decreased blood oxygen levels, this sensation will be picked up by your kidneys, and the kidneys are what produces this hormone called erythropoietin. And erythropoietin tells your red bone marrow and your bones to produce more erythrocytes. If we have increased erythrocytes, this causes an increase in blood oxygen levels. So whenever the body notices a decrease in oxygen levels, your kidneys will secrete this hormone erythropoietin, which will then tell your red bone marrow to stimulate the production of more erythrocytes, which means there will be more blood oxygen levels as those erythrocytes carry the oxygen to the tissues. So here we have a schematic graph of this. Decreased blood oxygen levels tells our kidneys to increase erythropoietin, that hormone production, which tells red bone marrow to increase the red blood cell production. And in turn, that will increase blood oxygen levels in the body. The fate of old erythrocytes and hemoglobin. So again, your erythrocytes only live for about 120 days in the body. And when they're old and dying, or they can't do their function anymore of transporting oxygen, they're removed from the blood by macrophages in your spleen and liver. The hemoglobin is broken down. The globin is broken down into individual amino acid components because remember, it's a protein. So we break it down into the amino acids. Hemoglobin's iron will always be recycled. And then the heme group, that molecular group that surrounded that iron, is converted to bilirubin. Bilirubin is taken up by your liver and is released into the small intestine as a part of bile. So it's kind of this idea of no waste in the body. Everything is used for a purpose. So we're eventually using that bilirubin, which is coming from the breakdown of hemoglobin, to create bile. And bile is important because bile will go into your small intestine to help degrade or destroy fats in the digestion process. So here we have hemoglobin breakdown. I don't think you need to know the specifics of this, but just the general idea that hemoglobin is broken down into heme, globin, which turns into amino acids. The heme groups turns into bilirubin, which goes to the liver. That eventually becomes bile, which helps your small intestines to degrade fat. And the iron can go into the liver, the spleen, or the bone to be recycled. Bilirubin will eventually also, if it's not used as bile, it will have derivatives that go into the blood, which are usually filtered out by the kidney in your urine. So then we get to leukocytes. So erythrocytes are red blood cells and leukocytes are white blood cells. So those are two of the three formed elements, erythrocytes and leukocytes. Leukocytes do not have hemoglobin and they're much larger than erythrocytes. They do contain a nucleus and their main function is to fight infection and remove dead cells and debris by phagocytosis, which again is like the eating up of these dead cells and debris. You have 5 types of leukocytes, and we divide them into either granulocytes or agranulocytes. And the granulocytes contain granules and include neutrophils, eosinophils, and basophils. And out of these 5 leukocytes, there might be a test question of matching, just really simply understanding the 5 types of leukocytes and what each kind of function is or what it looks like. Neutrophils are the most common. They remain in the blood for 10 to 12 hours before they move to tissue, and they are phagocytes. So they will eat up dead tissue cells or debris to get rid of them. So that's the neutrophil. Eosinophils help to reduce inflammation. They destroy parasites. And basophils are the least common. They help to release histamine and heparin, which is involved in the immune response at the site of an infection. Agranulocytes, so A means without, so they do not have specific granules or specks in them. Monocytes are the largest sized white blood cells and they produce macrophages. And lymphocytes are a part of the immune response that have several different types. So your lymphocytes will go on and produce T cells and B cells, which have specific functions. And it's your lymphocytes that lead to the production of antibodies in the body. So here's a look at the type of white blood cells. You have the granulocytes on the top, the neutrophils, eosinophils, basophils. The monocytes are this one. It usually looks like a very large kind of simple circle, and then this is a lymphocyte. Platelets then are minute fragments of cells, each consisting of a small amount of cytoplasm surrounded by a cell membrane. They're produced in the red bone marrow from very large cells called megakaryocytes. And what platelets are is when small fragments kind of break off from those megakaryocytes and enter the blood as platelets. And the platelets are what play an important role in preventing blood loss when they form these clots. Blood loss. So this occurs whenever you get a cut or a splinter, if a blood vessel is damaged. So the only way you bleed is when you cut through or damaged an artery or a vein. Blood can leak into other tissues. It can leak to the external environment, the internal environment, and disrupt normal function. And blood that is lost must be replaced by the production of new blood or by a transfusion. So if too much blood is lost, a transfusion must occur where you get blood from another source. In order to prevent blood loss, our body kind of goes through 3 steps. The vascular spasm. So think spasm, kind of a constricting or a movement. This is the temporary constriction of a blood vessel to try to kind of cut off the area of blood flow to where the damage has occurred. The platelet plug means this is when platelets try to seal up the small breaks in the vessel. And then blood clotting, which is also known as coagulation, where an actual blood clot forms at the place of damage. So vascular spasm is the immediate but temporary constriction of a blood vessel. Smooth muscle within that vessel contracts, and this constriction kind of closes that small vessel off to stop blood flow through them. A vascular spasm will be stimulated by different chemicals released by cells kind of the damaged blood vessel wall and also by platelets. The platelet plug formation is very important in maintaining the integrity of the damaged blood vessels. The formation of a platelet takes a series of steps, but in actuality, all this happens at the same time. Platelet adhesion occurs first when the platelets stick together or stick to the exposed collagen in the damaged blood vessel wall. And as they stick to that collagen, they become activated, change shape, release more chemicals. And as more and more platelets kind of aggregate together, fibrinogen forms, bridges between the fibrinogen receptors of numerous platelets, and this results in the platelet plug. Fibrinogen helps to form bridges with the fibrin proteins to kind of help stick everything together. And I'm hoping we'll eventually show a picture, but this just shows platelets adhesing to kind of the area of damage. So the platelets adhere or stick to the exposed collagen of the blood vessel walls. And that releases different chemicals, which kind of encourages these fibrinogen fibers to bind to fibrinogen receptors on those platelets that are sticking to the damaged blood vessel wall. So we kind of get this spider web kind of structure that's forming to help clot or stop blood from leaking out of a blood vessel. So kind of interesting there. Blood can be transformed from a liquid to a gel, and this is what we call a clot. So a network of thread-like fibers called fibrin can trap blood vessels in fluid, and this will create a clot. It depends on a number of clotting factors. So different proteins in the plasma that are only activated following an injury that are made in the liver and require vitamin K. Steps specifically in clot formation. First, we have an injury occurring to a blood vessel. This causes an inactive clotting factor to become activated due to exposed connective tissue or release of thromboplastin. Prothrombinase, which is a clotting factor, is formed and acts upon the prothrombin. This is then switched to an active form called thrombin. Thrombin activates fibrinogen into its active form fibrin, and fibrin is actually what will form the network that traps the blood or clots the blood. So here we have clot formation. Again, I don't think you need to know all of these steps and what is active, what is inactivated, but just know that it starts with an injury to a blood vessel, different chemicals are released, and basically we're activating clotting factors, which will then eventually activate fibrinogen to form our fibrin clot. And I wanted to see, I don't think it's going to show, there's usually sometimes often a good picture showing this, but maybe when we'll get a little bit farther. Clots need to be controlled so they don't spread throughout the body. So if a clot goes on to spread in the body, it can form blockage in a blood vessel. If this occurs in the lungs, it's very scary because it could block oxygen being delivered to your tissues. Clot blockage in the brain could result in a stroke. So we don't want clots to spread. So we have in our body anticoagulants, which prevent clots from forming. And examples of an anticoagulant is heparin and antithrombin. Injury can cause enough clotting factors to be activated that even anticoagulants can't work in that particular of the body. So if there is an injury in a particular area of the body, these anticoagulants will be overdrive. So they won't be able to do their normal function because we'll want clotting factors in that injury area. Once the kind of injured blood vessel has stopped bleeding, we have the clot retraction. So the clot condenses, serum in the plasma is squeezed out of the clot to help enhance healing. Fibrinolysis is the splitting of fibrin, helps to dissolve the clot, and plasminogen, the plasma protein, breaks down the clot itself. Good question, Matt. I'm not sure. Major differences in anticoagulants and platelet aggregation inhibitors. I would have to look into that. That's a good question. I would assume the function of the both would be similar, but they might have a different way that they function. Because one is inhibiting specifically, the aggregation or the platelets sticking together. Anticoagulants might have a little bit different function, but that's a good question. I would have to look into that. So here's fibrinolysis. So we're trying to lyse the fibrin or break apart the clot. We have more activators coming in so that we're taking our fibrin and basically just breaking it down. So that's fibrinolysis. This word lysis always means splitting. So this literally means the splitting of the fibrin or the splitting away of that clot. So now we'll talk about blood grouping. And this is important to know because if you ever are injured very severely, you might need a blood transfusion or a donation of blood from someone else. A transfusion reaction can be a bad reaction if it causes agglutination, which is when the blood cells clump together. So there's a bad reaction between a donated and a recipient or a receiver blood so that they clump together because they're not compatible. And agglutination means a clumping of blood cells, and this is always bad. We have to define a few key words before we talk about blood groups or blood types. Antigens are molecules on the surface of your red blood cells, your erythrocytes. Antibodies are proteins in the plasma. And blood groups are always named according to the antigen. So for example, I am type A blood, so I have A antigens on the surface of my erythrocytes. If you're type B blood, you have B antigens on the surface of your erythrocytes. You could be type AB blood, where you have both antigens on the surface of your red blood cells, or you could be type O blood, where you have neither A and neither B antigens on the surface of your red blood cells. To add in another fun thing, and we'll go over this, you can be positive or negative. And this positive or negative refers to the presence or absence of another antigen called the D antigen or the Rh factor. And if you're A positive, you have the A antigen on the surface of your red blood cells, and you have that D antigen or the Rh factor. If you're B negative, you have the B antigen on your red blood cells, but you don't have that Rh factor. I'm A positive, which makes up the majority of the population. So I'm I wouldn't say normal, but very common. I have a very common blood type, and we'll go over some of the other blood types in a little bit too. So this kind of just describes what I just told you. What blood type can be given to anyone? That's type O negative. That's the universal donor. So in your ABO system, there's two types of antigens that can appear on the surface of red blood cells, the type A antigen and type B antigen. If you're type A, like me, you have A antigens on the surface of your red blood cells. Type B blood has type B antigens, and type AB blood has both types of antigens. So if some of you are AB, you have both A and B antigens on the surface. If you're type O blood, this has neither A nor B antigens. So type O blood doesn't have the A and it doesn't have the B on the surface. The types of antigens found on the surface of your red blood cells can actually be genetically determined. So you get your blood type from your parents. They pass that down into kind of a gamete or a genetic square. So you can kind of, most often you'll have a blood type of one of your parents. So what are antibodies? So again, antibodies are in the plasma of the blood, and they are against the antigen that is usually present. So if you have plasma from a type A blood, you have anti-B antibodies, which act against the B antigens. So what this means, again, I am type A blood. So I have the A antigen on the surface of my red blood cells, and in my plasma contains anti-B antibodies, meaning my blood, I never want to receive blood from a type B person because I have anti-B antibodies that would attack that blood. So anti-B antibodies will act against type B antigens, so I could never receive blood from a type B person. Plasma from type B blood, so let's say Sally in our class has type B blood, so she has the B antigen on the surface of her red blood cells, and her plasma contains anti-A antibodies, which act against the A antigen. So basically, whatever your type of blood you are, you contain antibodies against the opposite type. So type B people could never receive blood from type A people because they would have antibodies that would attack that type A. Type AB blood is special. So type AB blood, so if you are type AB, you're kind of special and you're lucky. It has neither type of antibodies because if you think about it, let's say this is a red blood cell and it has A and B antigens on the surface, it won't contain any antibodies against itself. So type AB blood people have both antigens on the surface, but they don't have either type of antibody. So type AB people is considered the universal recipient. They can receive blood from anybody because they don't have any antibodies that are going to attack these antigens because they wouldn't have antibodies that will attack its own antigens because it has both on the surface. And type O blood plasma, remember type O blood has neither antigens on the surface. So because of that, type O blood plasma will contain both types of antibodies because it won't be attacking itself anyway because it doesn't have those antigens. Type O blood plasma is considered the universal donor. When we donate blood, we're donating red blood cells, not the plasma. So often the question is, well, type O blood people have antibodies that could attack, whoever they're donated to. Well, when you donate blood, you're donating just the red blood cells. You're not donating the plasma that has the antibodies. So that's why type O blood people, they can donate their blood to anyone because they don't have any surface antigens that would react with antibodies. Who's confused yet? All right, I think we'll eventually get into a chart that'll describe a little better here. In Caucasians, so for some reason, ABO blood groups are broken down based on the race. For some reason, some races have more of a group type than others. We don't know why, but in Caucasians in the United States, the distribution is actually, most is type O, which is 47%, type A, which is 41%, type B is 9%, and type AB is about 3%. So again, type AB are kind of the lucky ones because they can receive blood from anyone. We have a lot of people who are type O blood who, that's considered the universal donor. So if the red Red Cross knows you're type O. They probably call you and ask you to come and donate blood. Any of us can donate blood. It's all good to donate blood, but especially if you're type O. Among African Americans, the distribution is just kind of similar, a little less of type A, type B, and type AB. You guys don't really need to know that, but it's interesting to see the different percentages and breakdown. So let's go back and talk about these antigens and antibodies, because usually I'm sure I've lost half of you by now. So if we look at, again, your red blood cells and your blood type. So let's look at type A blood. And again, I pick on myself because I'm type A. So again, my red blood cells have the A antigen attached to the surface. And you'll notice that type A antigen has a different shape to it. In my plasma, I create anti-B antibodies. So I will create antibodies against the type B antigen. If we look down here at type B blood has the kind of triangular shaped B type antigen. And just look at it this shape for a second. The B antigen fits in really nicely to this anti-B antibody. So let's again say that I was donated type B blood. If I get type B blood, what happens is my anti-B antibodies, because they fit in very nicely, they'll come along and they'll grab hold of those antigens, and my antibodies will clump those antigen B red blood cells together. And that clumping of red blood cells, because my antibodies can grab hold of those antigen, that clumping is what causes agglutination, which means a bad transfusion has occurred. If your red blood cells clump together, that means they can't travel throughout the body to bring oxygen to tissues. So bad transfusions in people will cause death because you have antibodies kind of grabbing hold of the antigen that shouldn't be there, clumping your red blood cells together and causing basically no oxygen movement. In converse to that, we have type B blood with the B antigens. You notice in the plasma, it has the anti-A antibodies with kind of the curved shape to it. And that curved shape will fit in nicely to clump or grab hold of A antigens if we ever mix those blood types together. Then you'll notice type AB blood has both types of antigens on the surface. So type AB blood has both types of antigens. It doesn't have any antibodies in its plasma. So again, if you are type AB, you should be so happy right now because you can receive blood from anyone because you don't have antibodies in your plasma that are going to cause clumping or grab hold of any other donated blood that comes to you. And if you're type O blood, you're kind of considered the hero because you don't have any antigens on your surface. So these little red blood cells, they can be donated to anyone because these red blood cells, no antibodies will be able to grab hold of them and cause clumping. And in your plasma, if you're type O, you have anti-A and anti-B antibodies. So type O is considered the universal donor. They can donate their red blood cells to anyone. However, if you are type O, you're a little bit unlucky in the fact that you can only receive blood from other type O people. And that's because in your plasma, you have antibodies against both types of antigens. So type O people cannot receive blood from type A because it has anti-A antibodies that would cause agglutination. You can't receive type B blood because your anti-B antibodies would cause agglutination. So hopefully that explains it a little. Type AB blood is considered the universal recipient, and type O blood is considered the universal donor. And that's because the type AB can receive blood from anyone because they don't have any antibodies that would kind of cause agglutination. All right, so this shows you kind of what agglutination looks like. Again, here's type A blood of a donor, the anti-B antibody, and the type A blood of the recipient. So basically, we're kind of giving type A blood away to another type A recipient, And this does not cause any agglutination because the recipient's blood has B antibodies in the plasma, but it cannot fit or hold on to those A antigens. So type A blood can be donated to another type A person. However, if you are donating type A blood, let's say you're donating type A blood, and again, these are the A antigens on the surface of those red blood cells, you're donating it to a type B person. Remember, the type B person will have anti-A antibodies in its plasma. So look at what happens. The agglutination occurs where the antibodies will cause clumping because it can grab hold of the red blood cells, grabbing hold of those A antigens. And when it does that, it clumps the red blood cells together. The clumping of red blood cells is called agglutination. It's not good. That means those red blood cells cannot go on and travel throughout the body to deliver oxygen to the places that need it. All right, so a little bit more about this. Type O people are considered the universal donors because they have no antigens. And again, when you donate your blood, you're donating the red blood cells. You're not donating the plasma that goes with it. So type O people, they will donate their red blood cells. And again, their red blood cells have nothing on the surface. So no antibodies in the recipient's blood will be able to grab hold of them. So O are considered the universal donors. If you are type O, please consider donating blood. Type A people can receive from other type A people or O. Type B people can receive from B or type O, because again, type O can be given out to anyone. Type AB blood can receive from A, B, AB. O should be in there too. But type O people can only receive from other type O blood. And then we get to the positive and negative. And the positive and negative just means the absence or presence of the Rh blood group. And Ra positive means you have Rh antigens present. About 95% of us have the positive. So about 95% of us are O positive, AB positive, A positive, or B positive. What's interesting about this blood group is unlike the other A and B antigens where you normally have antibodies in your plasma that are present to fight off A and B, antibodies can only develop in an Rh negative person if it's exposed to Rh positive blood by a blood transfusion or from a mother to a fetus. So this is important in pregnancy, and I'll explain about that. So basically, if you are A negative, so you don't have the Rh antigen, you don't naturally have antibodies against the Rh factor unless you're exposed to Rh positive blood. And this is one of the common kind of issues in pregnancy. If a mother is Rh negative, so it does not have that Rh antigen, and the fetus is Rh positive, the mother can be exposed to that fetal blood through the placenta when it mixes with the mother's blood. When this happens at the first time, the mother's blood will start to produce antibodies against that Rh antigen. So again, you have a negative mother, but her fetus is positive, most likely that came from the dad. What happens the first time that blood mixes in kind of the first pregnancy, there's no real concerns because the mother will make antibodies against the antigen, but usually this occurs at birth, so that first fetus usually isn't formed. But if she has another pregnancy, so let's say her second pregnancy, her fetus is Rh positive again, she now has the antibodies built up against that factor. And that could cause what we say hemolysis of the newborn or hemolytic disease. And this occurs when those mothers' anti-Rh antibodies can cross the placenta and cause agglutination or literal splitting of blood cells in fetal red blood cells. And this could be fatal to the fetus. So this is why we would treat the mother with a shot called Rogam, which contains antibodies actually against the Rh antigen. So here we have hemolytic disease of the newborn. I think this is showing in pregnancy number 1 before or during delivery. Rh positive red blood cells from the fetus can enter the bloodstream of a Rh negative woman, usually through a tear in the placenta. And usually this occurs during delivery or right before. So, the mother would now form antibodies against the Rh factor, but it wouldn't affect the first delivery or the first baby. But in subsequent pregnancies, if the fetus is Rh positive again, then the mother's antibodies could circulate. The antibodies can cross the placenta, and those antibodies could get into fetal blood capillaries. And if those antibodies against the Rh factor in an Rh positive fetus, that could cause agglutination or clumping of fetal blood cells, eventually leading to lysis or splitting apart of those red blood cells, and that can be fatal. Okay, any questions so far? How are we doing, guys? Okay, if you're a little confused still, I would even, I recorded this lecture and maybe listen to me explain it again, or YouTube blood types, kind of to get another way of, describing blood types. But this is always one of the harder concepts to understand, but it's really simple when you think about it. Your blood type is just based on the antigen on your surface, and you produce antibodies against the opposite antigen. You just can't mix the blood types. It's not as simple, but hopefully what I said kind of makes sense, and do a little more research on your own and ask me more questions if you guys have more. So we use different blood tests as a diagnostic tool because the levels of your red blood cells and your white blood cells is important for being a precursor for disease. A complete blood count provides information such as red blood cell count, hemoglobin, which is the protein that carries oxygen, the hematocrit and white blood cell count. And the hematocrit is the percentage of the total blood volume composed of red blood cells. And if the hematocrit is lower than it should be, that means the percentage of your blood of red blood cells is lower than it should be. So that could affect oxygen delivery to tissues. Hemoglobin, this diagnostic blood test is important because to determine the amount of hemoglobin in the blood is extremely important, because hemoglobin is what transports oxygen to your tissues. And it could indicate anemia, which would be low oxygen levels. So we can withdraw blood into a hematocrit tube. Usually this can be just a prick. And again, we centrifuge it down to kind of separate our blood into the different components. Remember, plasma is always at the top, and red blood cells sink to the bottom because they're denser. And we can have the Hematocrit Scale. So the Hematocrit Scale, again, is the percentage of a blood sample that is red blood cells. In males, it's a little less than 50%, and in females, it's a little less, about 45%. The prothrombin time is another good diagnostic blood test. This is the time it takes for your blood to begin clotting, which is good to think about in terms of if you ever get damaged to a blood vessel. And the white blood cell count is usually an important one. It's the total number of white blood cells. And if your white blood cell count is extremely high, it usually means your body is fighting off disease. But if it's extremely low, it means your body cannot or won't be able to fight off disease. So the sickle cell anemia is a genetic condition that affects the shape of your red blood cells. And I don't think I go through that here. Let me, I just have 2 more slides to get through, and then I'll show you a picture of what sickle cells look like. They look sick. Yeah, moon-shaped is a good way to describe it. And yeah, it's not good, sickle cell anemia. So the white blood cell differential count, we can actually determine the percentage of each 5 kinds of leukocytes, because remember, each of these 5 leukocytes have a different function in the body. So we want the ranges of these leukocytes to be in a normal range. And if one is out of range, too little or too much, that could be another diagnostic sign for disease or that something is wrong. Leukopenia, enia means usually too little. This is a low white blood cell count. This can be caused by radiation or chemotherapy drugs that are killing off your cancer cells. And when they do that, they're killing off your white blood cells as well, also caused by tumors or a viral infection. Leukocytosis usually is a high white blood cell count, and usually your white blood cell count is extremely high if you are fighting off an infection or leukemia, which is a type of cancer in the blood. All right, so I'll go ahead and stop the recording.